Mesdames et messieurs, chers collègues, bonjour. On the behalf of the Department of Agriculture in the United States, I'd like to congratulate you for this fantastic and successful conference. And in particular, I'd like to thank Elizabeth Erlasher-Vindel and the Directeur Général of the OIE, Monique Eloise, for including a section on research in this important conference. And the reason is, for the last 2 days, we've discussed the challenges that we face with antimicrobial resistance, and we've talked about some of the problems associated with AMR, and perhaps some of the solutions, including the restrictions on the use of antibiotics. But when we look at our veterinarians and farmers, if we restrict the use of antibiotics, which are so important in the practice of veterinary medicine, what are the alternatives? I think I need some help with my presentation. Okay. Thank you. So after a brief introduction, I would like to give you and leave you with some information on alternatives to antibiotics. Not only a definition, but a few examples. And then I want to talk about antibiotic alternatives for growth promotion, the next steps, and then I'll have some concluding remarks. So we must not forget that with the discovery of penicillin, Alexander Fleming, who received the Nobel Prize in 1945, in his speech at the Nobel lecture, he already told us that he was already seeing resistance to penicillin. So before penicillin was ever commercialized, we already knew that antimicrobial resistance existed. If you look at the diagram on the left, it identifies some of the major antibiotics that were discovered, and inevitably, with time, the discovery of antimicrobial resistance was discovered. And of course, if you look at the diagram on the right, you see and we understand that the spread of antimicrobial resistance is associated both in animal settings on the farm as well as the human setting in hospitals, and that maybe those two settings converge. Antibiotics use in animals has been really critical, not only for therapeutic use, prophylactic, or even metaphilic use. And yes, we did use antibiotics for growth promotion. Regardless of your personal opinion, it's clear that there's an increasing concern due to antibiotic resistance, and there's a need for increased regulation and the restriction of these antibiotics. So let's examine, what are the alternatives to conventional antibiotics, which is definitely warranted. So alternatives to antibiotics. If we go back to the first symposium that the OIE organized in 2013, there were a series of recommendations. And recommendation number 13 specifically said that we do need to support relevant research, and in particular to find alternatives that could be used in animal production for antimicrobial agent substitutions. So what are these antibiotics? Let's first look at the definition of antibiotics. As we know, they can be used for, as defined by the OIE in the manual, the terrestrial code, they can be used to treat, to control, and to prevent animal diseases due to bacterial infections. So essentially, when we look at these alternatives, they have to achieve the same. If you look at the website that we started to organize as a resource for more information on alternatives to antibiotics, the specific definition that is provided is any substance that can be substituted for drugs that are increasingly becoming ineffective against pathogenic bacteria, viruses, or parasites. Yes, viruses and parasites, because as we heard yesterday, we also have challenges with antivirals and resistance, as well as antiparasiticides, or even acaricides for pest controls of ticks. So this is the challenge that we have, and research needs to play a fundamental role in finding some solutions. Now, back in 2012, we organized the first symposium on alternatives to antibiotics with the support of the OIE. And the objective of that symposium was to highlight some of the promising research results on alternatives to antibiotics, assess the challenges associated with their commercialization and use, and provide actionable strategies to enable the development of alternatives. Interestingly, the three main conclusions from that first symposium was that although we knew that there was already the sale of a lot of these alternatives to antibiotics, such as feed additives, very similar to what we see on the human side, where today hundreds of different nutraceuticals are being sold. But the problem is there's very little scientific information on their mode of action and use. And so clearly from that first symposium, one of the things that we learned was that there was insufficient research to really support their effective use in animal agriculture. Second, alternatives to antibiotics must be developed according to national and international standards and meet requirements for efficacy, safety, and quality. The good news, though, the regulators that participated in the conference, regulators from pretty much all over the world, including the FDA and the EMA in Europe, identified and told us that actually regulatory pathways were in place to enable the registration of these alternatives to antibiotics. So how are we going to then provide the resources to actually develop them so that they can actually be used as alternatives to antibiotics? In 2016, we wanted to take a pulse of advances that have been made in research, and we organized a second international symposium, again with the support of the OIE, the European Medicine Agency, and the FDA. And what we focused on were 5 product categories. Vaccines has already been covered by Jean Moulin, but we also covered microbial-derived products, phytochemicals, immune-related products, and innovative drugs. Because of time constraints, I just want to give you some examples for 3 of them. Some of those are probably ready for application should companies wish to actually develop them and perhaps go through a regulatory process to ensure, again, their effectiveness, their efficacy, and quality. While some of them may be upstream, as we say in the pharmaceutical industry, but nevertheless, today we are in the 21st century. And we're making breakthrough innovations, and we have tools that are going to be available to us to essentially change the paradigm on how we look at anti-infective. So let's look, as an example, microbial-derived products. First of all, everyone in this room has heard about probiotics as antibiotic alternatives to improve gut health. In fact, some of you may have taken your probiotics this morning. Yesterday, we heard about this phenomenon called the microbiome. The question is, though, there's 2 extremes when it comes to probiotics. There's the use of a single probiotic, such as lactobacillus, to improve our health, where on the other extreme, there's some practices both on the human side and even consideration on the animal health side of doing total fecal transplants. But at the end, how do these probiotics actually work? And what's fascinating is that we're starting to understand the multiple role that they have in gut health. Anywhere from a competitive exclusion mechanism to actually providing substances that are anti-infectives and interaction with the epithelial cells that line the gut to modulate our immune response and lower inflammation. So the question is, when we look at the multitude of what we call commensal microorganisms in the gut, what are the families of microbes that can actually be substituted or even added to our food for a beneficial effect to essentially prevent or even control enteric pathogens? And that research is currently occurring, and in the future we're going to see more and more of these probiotics with defined mechanisms that can be used effectively to improve gut health. Some of you may have heard of this concept now that's made a lot of news in science recently called gene editing, thanks to the discovery of this CRISPR-Cas9 system. Essentially here, the idea is, well, we know that we have bacteria that have these antimicrobial resistant genes, but what about if we could actually effectively target those genes and make a bacteria that is resistant now not resistant by actually taking this CRISPR-Cas9 system, which is comprised of taking a bacteriophage, which are viruses of bacteria that target very specific pathogens, and actually delivered this enzyme and a nucleotide to actually mutate and inactivate this antimicrobial gene. And this, ladies and gentlemen, is actually this technology available to us today, which is really fascinating, and I think you will see many more applications of this technology in the future. Looking at phytochemicals, which is really critical. In our diet today, we know that there's more than 10,000 phytonutrients. We've heard of herbal medicine, and in fact, for thousands of years in Asia, phytochemicals have been used for medicinal purposes. The question, though, is how do they work? In veterinary medicine today, we're seeing a lot of companies that are already selling phytochemicals and phytonutrients as alternatives to antibiotics with all sorts of claims. But understanding their mechanism of action is going to be critical. And here before you, you have a list of 10 functions that have actually been defined through research, which includes everything from certain phytochemicals having a direct role as anti-infective, modulating the immune response, or even epigenetic effects, which is also a new phenomenon where now we understand that certain things can modify our DNA and affect gene expression. The problem is, even though we know they have some effect, there's this concept of hormesis where we know we can have a beneficial effect of some phytochemicals, but there's also can be a toxic dose. And so research and taking some of these products or substances through a regulatory process so that they can be properly developed to ensure, again, their safety, efficacy, and quality is going to be critical. And we're really at the stage now where we can go from a black box to both sides. Now that we have access to the animal genomes and we can look at the transcriptomes, in other words, all of the expression of these genes by measuring messenger RNAs, as well as the proteomes, all of the proteins that are associated with this, we're actually able now to measure the direct effect of some of these phytochemicals, including what genes are upregulated and what genes are downregulated in response to some of these phytochemicals. And this diagram before you actually identifies some experiments that have been done in poultry for a disease called necrotic enteritis, which in fact is becoming pretty prominent in the poultry industry by removing antibiotics. And what you see in these bar graph on the right is the measured designation of the different microbiome, all of the bacterial species that are in a gut, and the three different groups essentially show three different genetic lines that are commonly used in the United States for poultry. And every one of them has a different set of microbiome. Their microbiome is actually different. And when we look at the effectiveness of some of these phytochemicals and the genes that they regulate, what was interesting is actually they actually work against this disease, necrotic enteritis, but it only worked in one of those genetic species of poultry. So this is to show that science is complex, but research is going to really be critical to actually define those mechanisms of action so that they can be properly used. Lastly, I want to talk about immune-related products. Now, here's a very simple technology, which is essentially taking advantage of the fact that chickens produce eggs, and in that egg yolk, there's literally tons of this IgY antibodies that are produced. And the question is, if we're able to immunize a hen against a specific pathogen and produce a lot of this IgY antibody, can then be processed and included in our feed. And so this is now actually being done, and some companies are actually selling these IgY antibodies as an alternative to antibiotics. And the advantage is that there will be no risk of resistance, no toxic residues, and it can simply be used. The last one that I'll present is the fact that through the genomes, we've discovered that our epithelial cells in the gut are actually, like bacteria, producing antimicrobial peptides. So can we capture and manufacture these antimicrobial peptides and actually use them as alternatives to antibiotics? And this example here is the discovery of NK lysine and the fact that they actually kill every apicomplexa, protozoans such as Cryptosporidium or coccidiosis, and in fact could be used effectively as an alternative to antibiotics. So I'm running out of time. I just want to briefly talk about antibiotics and growth promotion, which has a bad connotation. But for farmers, what is going to be the substitute? Now, when you look at the mechanisms of antibiotics as growth promotants, we actually never really fully understand how they function. And that's going to be critical if we are to find these different alternatives that can essentially replicate those different mechanisms of growth promotion. I want to take you back with this slide to this gut microbiome, and the fact that the gut microbiome in the gut is connected to every organ in our body, and is critical in terms of our central nervous system, as well as the modulation of the immune system, as I previously stated. And one of the things that we discovered that's associated with those effects is the thousands of metabolites that are actually produced and are part of some of the physiological effects that we see in our well-being and our fight against infections by bacterial and viruses. What's always been interesting to me is if you look at this diagram, using some of the latest technology of mass spectrometry, this study looked at human serum, and they identified that in human serum, we actually have over 7,000 metabolites that are produced. The thing is, when you go to the doctor, hopefully most of you have had your annual checkup, you probably are given, when you take your blood test, 35 compounds. Your lipids, your potassium, your chlorophyll, your chlorides, enzymes, but there's 7,000 metabolites, most of which we don't know their function. Where they came from, are they produced by our own cells, or are they produced by those commensal organisms that we have in our gut? So now we have the tool to, some of you may have heard, the metabolome, to actually measure those metabolites in experiments. And in this study here, the question was, well, what metabolites are actually upregulated or downregulated in response to the use of Virginiamycin and bacitracin that, as you know, were used as growth promotants. And in fact, 199 metabolites were upregulated in response to the use of these antibiotics, many of which are associated with these CAG codes, which are specific biochemical pathways that are associated with our physiology. And so going forward now, we really have the means to actually understand perhaps those mechanisms by which antibiotics provide a growth promotion. And then can we find alternatives to actually replicate those mechanisms? So very briefly, in terms of the next step, one of the things that we wanted to do in 2016 is to actually provide peer-reviewed publications around these alternatives to antibiotics. And we accomplished that task. What you have here is a list of review articles that essentially summarize some of these technologies, whether they're vaccines or microbial-derived products, phytochemicals, again, anything that's related to the immune system, as well as innovative drugs, chemicals, and enzymes. And so if you're interested in this, I encourage you to pull these articles and read them. I think you will find them very interesting. And importantly, we also achieved, working with regulators, a publication that's entitled Regulatory Pathways to Enable the Licensing of Alternatives to Antibiotics, which is going to be really critical because we really have a gap and a lack of incentive for the pharmaceutical industry to actually invest in the development of these alternatives to antibiotics. One of the things, an organization that's going to be very important in solving that problem is an organization called STAR-IDAS, which is better known as the International Research Consortium on Animal Health. And what they are doing is a series of gap analyses to understand where the gaps are in research. And in context of AMR, which they are going to tackle, what are the gaps and what can we do to fill the gaps and promote research and fund research, and somehow link research funders to target priority research gaps to advance the development of alternatives to antibiotics. And this is going to be very important in the future for the implementation of that research. And lastly, we are going to organize a third international symposium on alternatives to antibiotics in animal production. That will take place in November in 2019 in Bangkok, Thailand. If you go to that ATA website that I referred, the information will be provided for this symposium, and we hope that many of you will be able to attend this important symposium where we're going to further dive into some of the new technologies that are being developed to address the issue of antimicrobial resistance. So in conclusion, I'd like to leave you with four main things, and one is going forward, I think it's going to be very critical to actually link academia, government researchers, pharmaceutical industry, the feed industry, regulatory agency, and livestock industries together in promoting and being champions of this change in paradigms and focusing on these alternatives to antibiotics. Second, as I've said many times during this presentation, defining the mechanisms of action of alternatives to antibiotic is going to be paramount to enable their effective use, whether they are used for prevention, treatment, or even enhance the health and production of animals. Clearly, going forward, we really need to look at science differently, and we really need to integrate nutrition, health, and disease research. And lastly, we may not be able to find the miracle drug to substitute everything that antibiotics do, but I think it's going to be important to essentially maybe look at a portfolio of alternatives to antibiotics that may be considered to achieve optimum health and disease management for different animal production systems. I'd like to acknowledge several scientists that have contributed to the organization of these scientific meetings that have focused on alternatives to antibiotics. Yoon Lily Hoy, a scientist in my organization at USDA ARS, Bruce Seal at Oregon State University, Chen-Bo Yang at University of Manitoba, Philip Van Immersel at University of Ghent, Karen Alser of the Pew Charitable Trust, Hank Hasman of Utrecht University, Elizabeth Elisha Vendel of the OIE, and Faye Ionu of the EMA, and Cindy Bernstein of the FDA, and many other scientists worldwide that are focusing in providing solution to this important issue of antimicrobial resistance. So with this, I thank you for your attention, and I wish you a pleasant, remaining successful symposium today. Thank you very much.